Sorry, where will these be actually posted so we can watch them again if we need to, please?
I think I would probably ask online to see where they put it. They should probably put it at the assessment section. But I'll find out where they put it, and then I'll.
Yeah, I couldn't find the other one that we did the other night today. So yeah, I thought I'd better ask. Thank you.
Okay, you're welcome. So this is our third and possibly our final exam revision, but that doesn't mean that you cannot touch base with me by email for one-on-one or by phone, so we could discuss this further. So it's BIOB, and I want to start by showing you the date for the exams. I'm sure by now everyone has downloaded the exam timetable. So this BIOB will be Thursday, 25th of June, and you would sit it at home with your computer. Hi, Sasha.
Hi, Eric. How are you?
I'm well, thank you. This is what your exams will look like. That's the front page, and the information that's important for you to be. This is chemistry, biochemistry, semester 1. So, and the duration for the exams is 2.5 hours. You'll be marked out of 100, but the actual weighting is 40%, which means that you have already done quiz 1, quiz 2, and the assignment, which is 60%. For some of you would have probably passed already. So this is not the time to be stressed at all. And you've also been given information what to do, what to study, what to review. And so that's exactly what we are going to go over today. There are 3 parts to your exams. The part A is multiple choice questions. This is 36 questions. It's almost similar to the multiple choice you've just done, quiz 1 and quiz 2. At this time, it's taken from session 15, from enzymes to the end of our metabolism, proteins. All you have to do, the best way to study for this is to ensure that all the tutorials that you've done between session 15 and session 25, if you were to come across it again, there's no way you should get it wrong. So I would like to practice again and again. And then there are also review quizzes as well that's very similar to the tutorials anyway, that you can go over and practice. It's the best way to study for some of us will be just go over again and again until you are confident, you are comfortable with it. So that would be the part A. It's if you get 1 point correct, you get 1 mark. So if you get 30 or 36 of them, most students do very well with this one, because you've done it in quiz 1, you've done it in quiz 2, and this is possibly more like quiz 3. The part B is matching blank. There are 2 parts to part B. First one, an example. I think I'll show you an example of part B, what it looks like. It's something like that, matching. The first part is 4 marks. For instance, you can be asked what is glycolysis, and then you look on the right side and select what you think is the best definition for glycolysis. And we look at this and we say that in glycolysis, we take glucose and we break it down into 2 parts. That's the best definition there. And then if you know glycogenesis is the of every time you hear of genesis, it's synthesis of glycogen. Genesis is synthesis. And say glycogen synthesis. And that will be a synthesis of glycogen. And so you select what you think is the correct response for that. And glycogenolysis, lysis is always the breakdown. When you say cell lysis or hemolysis, that's the breakdown of cells. So glycogenolysis is the breakdown of glycogen, the glucose. And so you look and select the one that you think is the breakdown of, which is A, breakdown glycogen, aerobic. Sometimes the matching questions are on the digestion part. For instance, if you are asked alpha amylase and pancreatic amylase, which one comes first? If you are to, which one would be the first to be used? And everyone knows that alpha amylase is where? In the mouth, and pancreatic is in the
Small intestine. Small
small
intestine.
So, yes. And so that would be amylase will be alpha 1 will be the first to be used. And if you look at lipid digestion as well, you have, what's the first enzyme to be used in lipid digestion? Anyone out there?
Is it lingual ligase?
Yes, lingual lipase. And what's the first enzyme to be used in protein digestion? Anyone know? It's pepsin. Pepsin. And so then you follow, if I know that this is alpha amylase or pancreatic amylase, and then the disaccharides. And then lipids, the same way. Yeah. And the next part is part of the part B is what no one should get any of this wrong. Why? Because you've already done it in your assignment. What this is asking you is if you've done all the pathways, then you already know the inputs, the outputs, and the name of the pathways. Who thinks they already have idea of all the inputs and the outputs as you see here on the pathway diagrams? Straightforward. All what I would probably advise is that when you are looking at the pathway, make sure that you know the direction of the arrow. Because sometimes students in a hurry to write the answers down or select the correct answer, they overlook where the arrow is pointing to. For instance, if glucose is the input and pyruvate is the output, what will be the pathway? If you look on the diagram.
Glycolysis.
Glycolysis, excellent. What happens if you see that the arrow is pointing in the opposite direction? Input is pyruvate and output is glucose. What will be the process or the pathway? And you know that it's what? Gluconeogenesis. The other one. Excellent. And then if you look at the first, if the input is acetyl-CoA, the pathway, that cyclic pathway that takes on acetyl-CoA, what will be the name of that pathway?
Citric acid cycle.
Citric acid cycle, excellent. Citric acid cycle. Now, what's the output for citric acid cycle? Who knows that? Output for citric acid cycle.
NAD.
FAD.
And FADH2, excellent. What happens between what happens to the NADH and FADH2? What is stripped of them? Electrons and hydrogen, and they are transported through this. And who sits at the end of it to accept the electrons and hydrogens? What molecule is at the end of the electron transport chain that picks up the transported electrons and hydrogen?
Oxygen.
Oxygen. And so sometimes you'll be asked, you see the diagram, it's asking you who's picking up oxygen and hydrogen, and you say, and the answer is oxygen. On the other hand, sometimes the oxygen is already sitting there and they're asking you, what does oxygen pick up in order to be, the arrow is pointing to water, in order to become water. And you know, it's picking up two things. What are the two things that oxygen picks up in order to become water?
Hydrogen and electrons.
Hydrogen and electrons, two hydrogens, two electrons. Excellent. Now, so we're quite familiar with glucose now. Now, this acetyl-CoA, how many pathways or how many processes can give us acetyl-CoA?
3.
Yes, we have, what's one of them? It will be what? Beta what? Oxidation. So you look carefully, yes, you look carefully where the arrow is pointing to, and you can see that input is fatty acids and output is what? Acetyl-CoA. If the arrow is pointing the opposite direction, that way and that way, then input is what? Acetyl-CoA, output is fatty acids, then the pathway is what? Lipogenesis. So as you review these pathways, make sure you look at where the arrows are pointing to and therefore what the names are. Now, what more can we say? We finish with, why is lipogenesis different from beta oxidation? Because a lot of students confuse that. What is lipogenesis? Anyone out there?
It's the formation from a non-carbohydrate source.
You are looking at lipogenesis. That is, if you are getting carbon skeleton from amino acids and lactate, and then you go that way to form fatty acids. Yes, but what is the name of when you have dietary lipids, whether it's in your digestive system or whether it's in your blod, and you split it into fatty acids and glycerol? What would you call that one?
Lipolysis.
Lipolysis. And you know that lipolysis is completely different from what? Beta oxidation. I want you to make sure that you don't confuse the two. Lipolysis is when you do it in the digestive system, or when the tags are in your blood or in your adipose tissue, and you split it up to use it. That is lipolysis. Is that clear for everyone?
Yeah.
Okay.
Yeah.
Now let's go to the dietary proteins. What happens once you break up proteins? What would be the final products of protein digestion?
Amino acids.
Amino acids. So what you will see in your exam is that all of this, some of it will be missing. Amino acids is missing, but if you know that from the diet you get amino acids, you select amino acids. And then you see that there is a transamination and deamination reaction. Two things come out of it. What would the first ammonium ions and the carbon skeleton? But if this input is going to go through the urea cycle, then you know that that is what? Ammonium ion. Finally, what's the output of the urea cycle?
Urea.
That one there. Urea. Any questions on how to answer this particular pathway? You have 8 of them missing, and all you are being asked to do is what are they? What is the input? What is the output? And what is the name of the pathway? And it's straight away, everyone should be able to get that. And that's a cool 4 marks. Is that okay?
Yep.
So it should be. All right. And so we'll move on. What's important now is you know how to answer part A, how to study for the part A, you know how to study for the part B. The part C, we've done some of them already, but I want to just go a little round and see what it is we need to do in part C. A whopping 56 marks. And there are 7 questions. And you are being asked to do A or B. Don't do both. One A or B. For instance, A could be citric acid cycle or gluconeogenesis. And you can see, oh, I think I like citric acid cycle, so I'll do that one. And the questions are set up exactly as they have been set up in your tutorial. For instance, what is the input for the citric acid cycle? And where do the input come from? Who knows this question? What is the input for citric acid cycle and where do they come from?
Acetyl-CoA.
Acetyl-CoA. So if you say the input for citric acid cycle is acetyl-CoA, you get 1 mark. And then if you look at the question critically, the second part of the question says, where do they come from?
So either carbohydrates, proteins, or lipids.
Excellent. You can get it from carbohydrates, pyruvate to acetyl-CoA, or you can get it as a carbon skeleton from what? Amino acid degradation, metabolism, or you can get it as what? Fatty acid or beta oxidation. And if you do that, you get a maximum of 2 marks. What you don't have to do, what you need to remember is that all of those questions, the more you explain it, the more you can get a maximum 2 marks. There are 6 questions. If you just answer acetyl-CoA, 0.5, but the substrate or the input for acetyl-CoA is for citric acid cycles, acetyl-CoA. And then sometimes you're asked, what would they be used for? What would acetyl-CoA be used for? Who can just say, what do you think they are used for?
To begin the citric acid cycle.
Begin the citric acid cycle to generate, if someone would say it begins as it's used to begin a citric acid cycle, and then finally to generate what? Coenzyme NAD and FADH2. If you say that, you get 2 points. And the person who just says that it's acetyl-CoA, it's used in the citric acid cycle, will get 1 mark. So look very carefully at how the question has been asked. And then because most of, you know the answers already, it's important that you put all what you know down. 2 points, 2 points, 2 points. The maximum you can get is 8 marks, which means that some students, some of the 1 to 6 questions, some students might not be able to answer some of them, but they will still get the maximum what? 8 points. Is everyone clear on that one?
Yep.
Okay. So what you do next is that these are in the exam guide. You've been given all the topics that you will see in your exams. What's important is that you go back and revise it and learn it. Once you know it, that's what's important, that you have learned this. And the first one is the role of enzyme. We've done this section here. So I'll just go briefly over it, and then we go back to the second section. If you were not here the other 2 days, all you need to do is go to the first question is session 15, question 3. Yep. Yes. And so you go to the tutorial or tutorial answers, and then you look at session 15, question 3, and then you review it. If in your revision you still do not understand it, send me an email for explanation or ask me to give you a call and explain it to you. Is everyone clear on this one?
Yes.
So for those who are coming for the first time, what question here do they think they would need further explanation for? So we can at least go to a couple of them before we go to the next session, this section. So if you're coming for the first time, what are these questions that you think you probably need help with? So we can go and look at it and discuss it as a group. Can I ask a few questions on my own?
Yeah.
Okay. What is the role of enzyme in chemical reactions? You still remember enzymes if they've done it?
Lower the activation energy.
Yes, it lowers activation energy. You get 1 point there. What
More
else would enzymes do?
They catalyze
Yes,
the chemical
go ahead.
reactions.
Yes, so if someone says these 2 things, they get 2 points. If you just say they lower activation energy, you only get what? 1 point. And if you say they catalyze chemical reactions or speed up chemical reactions, you get the mark as well. Is everyone clear on that one? What about enzyme inhibitors? If you want, why do cells need to inhibit enzymes sometimes? That's not the question. The question is, there are competitive inhibition, non-competitive inhibition, and uncompetitive inhibition. Well, what's the role of competitive inhibition? Anyone remember that one? What are they competing for?
The active site.
The active site. So competitive inhibition will bind to the enzyme active site, and then you go on and say that by binding to the active site, it prevents what? The substrate from what? Coming in, and so it's actually
Binding.
Yeah, it binds and then prevents substrate from coming there, and so it inhibits the reaction. You get 2 points. What about non-competitive inhibition? Who still remember that one? Where do they bind?
To the regulatory site.
The regulatory site. And once they bind to the regulatory site, you get, if you say exactly what you have said, you get 1 point. What else happens once they bind to the regulatory site?
The shape of the active
shape.
site changes.
It change the active site. By changing the active site, it means the substrate cannot what? Bind. Does that make sense for everyone?
Yeah.
Yeah.
Okay. And then if you are asked, what is the actual process of the enzyme reaction? And then you look at it and say, oh, what happens when there's an enzyme reaction? First, the substrate has to bind to where?
The active site.
The active
The active
site.
site. And once it's bound to the active site, what do we call it? Enzymes.
Enzyme
Enzyme
substrate
substrate.
complex.
Yes, and then the reaction takes place and a product is what? Released. So you
Released.
follow a sequence. Yes, and then you get your full marks there. Now, carbohydrate digestion. Who still remember anything about it? You wrote it in your assignment.
Yep.
Begins in the mouth.
What happens? Yes, what happens in the mouth?
Alpha amylase enzymes break down polysaccharides.
What bond? Yes, what bond does it break?
Alpha glycosidic bond.
Glycosidic.
Excellent. So you need to mention these things in order to get all the marks. And once that happens, anything happens in the stomach?
No.
No.
No. No. And then once it goes into the small intestine, what happens there? What enzyme first acts on it?
Pancreatic.
Amylase. Alpha amylase.
Amylase. What does pancreatic amylase do? Exactly the same thing you said about the first one.
Breaks glycosidic bonds.
For the third one. It breaks the glycosidic bonds, releasing what? Smaller one.
Disaccharides.
And then what about lactase? What does lactase do? You still remember lactase?
It breaks down
The
the
enzyme.
lactose disaccharide.
Yes.
Yes, and remember to bring in the bond that is breaking. Is that okay? Because once you say it breaks the glycosidic bond between glucose and what? The lactose, and then you get the full marks. Now, let's come to glycolysis. What's the input for glycolysis?
Glucose.
Glucose.
Glucose. And where does the glucose come from?
Dietary carbs.
Digestion.
Digestion of what?
Carbohydrates.
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 Excellent. So if someone's looked at it and said, what is the input for glycolysis? And then not look at what other question is following it. So you get 1 mark there. But the person who looks at it and said, glycolysis, the input is glucose, and the glucose is coming from what? As the product of what? Digestion of carbohydrates. You get a two marks. And what happens to the glucose in glycolysis? What happens to the glucose in glycolysis?
It's broken down into pyruvate.
How many?
Oh, it's moved into the cell.
Into 2 pyruvates. That's exactly what you got it right. What happens to the glucose converted into 2 pyruvates, and then the pyruvates it's changed into acetyl-CoA, so we get energy out of it. Then you get all the marks there. So what I'm trying to point out to you is that to do very well, make sure that you look at the question critically, and make sure you add the extra points that they're asking for. And what is the outcome? The pyruvate. What happens to the pyruvate from glycolysis? What happens to it?
It's oxidized into acetyl-CoA.
Yes, and you get 1 point, and then you can say that the acetyl-CoA from it is then used in citric acid cycle to generate what? More ATP, more NADH and FADH2. So, yes, so put the links in to show that you know your stuff. Does that make sense to everyone?
Yeah.
Yep.
All right. Now, citric acid cycle. What is the input there?
Acetyl-CoA.
Acetyl-CoA.
And what happens to the acetyl-CoA in the citric acid cycle? What happens to it? Converted into? What do we get out the final output of the citric acid cycle?
Electron-rich coenzymes.
Electron-rich coenzymes, and you mentioned them, NADH and what?
And FADH.
FADH2, which would be used in what? The
Electron transport chain. Unoxidative.
So remember to, yes, excellent. Everyone's clear on these ones?
Mm-hmm.
So all you have to do now is just take one at a time, go back to the tutorial answers, look at session 15, question 3, and answer them. Is everyone clear on that one?
Mm-hmm.
Now, next question. Electron transport chain, what is the input for that one?
The NADH and FADH.
Yes, and what happens when at the, what happens to the input?
The
What happens to the-
hydrogen and electrons are transported.
Stripped off? Yeah, stripped of them
Yeah.
through redox reactions. Yeah, they are taken off. You can use any words. They are taken off by the protein complexes. Some people would just directly say FMN. And so it's the electrons and hydrogens are stripped off and then transported. The electrons are transported across what? The protein complexes. Another question that you can be asked, or from your note, from your assignment. The main reason you did the assignment is to try and help you understand the pathway so you can answer your exam questions as well. So if you did your assignment well, you find this extremely straightforward. Now, next question. From complex 1 to how does electrons move from complex 1 to complex 3? Who knows that?
Enzymes, CoQ and something else.
Coenzyme Q. Yes, coenzyme Q transports, which is a mobile electron carrier, carries electron from what? That one. What about from such complex 3 to complex 4? How are electrons carried across the two protein complexes?
With cytochrome C.
Cytochrome C, excellent. You can say electrons are carried by the cytochrome C and carried to. Finally, what is the final electron acceptor for those electrons and hydrogens?
Oxygen.
Oxygen. And what happens to it? You can say oxygen is the final electron acceptor, accepts the electrons and becomes what?
Water.
Yes. Is that clear for everyone?
Mm-hmm.
Now, another question. What the complexes? How does ATP? How do we get ATP from oxidative phosphorylation? How do we get it from there?
The energy of the hydrogen coming down through ATP synthase helps to bind ADP and phosphate together to create ATP.
Okay, that's excellent. And ATP producing ATP from metabolic pathways. When you combine ADP and phosphate, do you need energy to be able to do that?
Yeah.
Yes or yes? What happens when you break phosphate off ATP? What happens? Do you release energy or do you need energy?
Release energy.
You release energy, and so that's excellent. And now let's have a look. Any questions? Because we did this the last two times. So if you don't have any questions here, or maybe I'll ask one more. Gluconeogenesis. What is the input of gluconeogenesis?
Non-carbohydrate sources.
Yes, pyruvate from
pyruvate.
non-carbohydrate. Yes, from non-carbohydrate sources. What's an example of a non-carbohydrate source for pyruvate? Where do we get that from? Apart from carbohydrate, where do we get the pyruvate from? You remember the carbon skeleton?
From amino acids. Protein.
Yes, amino acids. The amino acids. If you take the amino group of amino acids, that carbon skeleton, some of them are pyruvate, some of them are acetyl-CoA, some of them are oxaloacetate. The next question you can be asked is what let's have a look. All right. If on the diagram, if you look at what will be the output for gluconeogenesis?
Glucose.
What do you get out of glucose? And then you go on, if you say glucose, you get 0.5. You say the output for gluconeogenesis is glucose, which is then released into where? Into the blood and then taken into other tissues. We just look carefully at the question, how the question has been asked. Output and what happens to the output. Is everyone clear on that one?
Mm-hmm.
Any more questions? All right, now the next one. hormonal control. This is one question that people, I've seen almost everyone get 8 marks. So if you don't get 8 marks on that one, it means you're losing. The first question is, when in the fed state, what exactly does that mean in the fed state?
That enough ATP has been produced.
Is that what that means? Yes, you produce enough ATP, but when someone says in the fed state.
When you're eating or after you've just eaten?
Yes, after you've just eaten an adequate amount of carbohydrate meal, which hormone is required and what happens?
Insulin.
Insulin, excellent. And the next question you'll be asked is, if you answer that question, then this is the way it goes. And where does the insulin come from?
Pancreas.
So how would you
Beta
so
cells.
if you say the insulin is released or synthesized or released by the pancreas, and then act on what? Glucose in the blood, so that the glucose is what taken into where?
Into the
Cells.
cell.
That's how you get, yes, that's how you get all the marks for that. Where the insulin is coming from, how it acts on the glucose, and where the glucose goes. What happens when you are in the fasting state? What hormone do you need?
Glucagon.
Fasting state. Glucagon. And so how will you answer that question? Knowing that you need to know where the glucagon is coming from, what it does.
Glucagon is released from the pancreas so that it can bring glucose out of the cells to be used for tissues and organs that need glucose.
Excellent. I will give you all the 2 marks for that question. Is everyone clear on that one?
Mm-hmm.
And so if you are not clear, all you need to do is go to session 21, look at question 5 and go over it again. Everyone's clear? We will do that shortly as well. And the next question I will ask is, so we've done the fed state. What metabolic pathways does insulin control? Activate or inhibit? That's the question you could ask, or that's the question there in session 21. What metabolic pathways does insulin activate or inhibit? Anyone else?
Glycolysis.
When you
Activate glycolysis.
Yes, what other ones? It activates 2 of them.
Glycogenesis.
Glycogenesis. Excellent. So you get 1 point there. And then what pathways does it inhibit? Insulin inhibit. If you have glycolysis, you don't need the others.
Glycogenolysis.
What's the opposite of glycolysis? Glycogenolysis, yes. And then what else? There's one more.
Gluconeogenesis.
Excellent. And so you've gotten all the 2 marks for that one. What about, then you go back to glucagon. What pathways does glucagon activate and inactivate?
So the opposite
And
of
you swap it
insulin.
around. Opposite. Yes, you swap it around. So you look what you did for the first one, and then you swap it around. Is that okay there?
Yeah.
All right. Now, lipid digestion. I think I'm going to go off to that question. Lipid digestion, session 22 and 1. Session 22. Yeah. And we go over it and look at it again. Because it's important. The digestion, all the digestions are important. So answer the following question. Does lipid digestion occur in the mouth? Explain why and why not. You see how that question has been asked. There are 2 parts to the question. So all the time, look for the first part. Once you finish, look for the second part. So that's how you get all the 2 marks. And if you say yes, it does occur in the mouth because there is a small amount of it by an enzyme called what? Lingual what? Lipase. Only a small part takes place there, very little part. If you are able to say this, then you get 2 points. If you say no, then you get no points because it does, there is a small amount Question 2. What role does the stomach play in lipid digestion? Are any enzymes present? If so, what do the enzymes do? Yes, the role is the stomach churn around, produces smaller droplets within the chine. Why do you think that is important? You're not asked that way though. If you're able to break fat into these little droplets, it allows the gastric lipase to digest it, because the enzyme is water soluble, lipids are not water soluble. So by churning it around and getting it into little droplets, it allows that. And what do the enzymes do? They start to break ester bonds within tag. So all you do is put this in 1 or 2 points, and then you get all the 2 marks. Everyone's clear on that one?
Mm-hmm.
Any questions? Matilda, are you okay?
Yeah, I'm okay, thank you.
Okay. Next question, pancreatic lipase. Let's have a look at that question there. What does pancreatic lipase do? Break down tag into MARCs and fatty acids associated to form micelles. What I want to ask you these 2 questions. If someone asks you the breakup of tags and the formation of micelles, which one happens first? From this question. You get the breakup, you get the like digestion first, and then once you get digestion, then you form what? The micelles, which allow the transport. Does that make sense?
Yeah.
It's not, you don't form the micelles first before you break it down. You break it down first, and then you form that micelle type. So it makes sense for everyone. Any questions? Because the reason why you need micelles, because of the intestinal environment is polar and lipids are non-polar. We need the formation of the micelles that allow it to be slightly polar, so we can just move it in the polar environment. Any questions?
No.
If you're okay. Which one forms first? The ester breakup or micelle formation?
The ester
Caroline?
break.
Yeah, we break up the ester links, and then afterwards we use the monos and the fatty acids to form what? The micelle. So we can, yeah. And okay, once the bloodstream of chylomicron releases tag, what is either stored or metabolized? Discuss it under what circumstances will tags be stored? When do you need to store tag? Is it fasting?
Third stage.
Or third stage. Excellent. Under what circumstances will it be metabolized? You can say that under what fasting state, or when all the glycogen stores are being used up, it would be then the body falls on lipids. Everyone's clear on that one?
Yeah.
And so that's all you do. You go into that question, and then you look at the results and revise it.
Sorry, what
question.
What are those questions? Where are they from?
Yep. Where are the, let me have a look.
They're from all our tutorials.
Oh, okay.
22.
Oh, okay. Yeah.
What question did you ask?
No, I just wasn't sure where
there was a question about where the acetyl-CoAs come from. Yep. And where would the acetyl-CoAs that goes through citric acid cycle, where do they all come from? There are 3 places they come from. The first one would be pyruvate into what? Carbohydrate into pyruvate and then to acetyl-CoA. Number 2, fatty acids through beta oxidation into what? acetyl-CoA. And then amino acid metabolism, which oxidative deamination splits of amino group from the carbon skeleton. Some of the carbon skeletons are acetyl-CoA directly, some are pyruvate, and some are acetyl-CoA. Would that answer your question? Okay. Now, beta oxidation. Let's try and answer a few questions there. Where do we go to get information on beta oxidation? Session 22 and
Missions 2 and 3.
2 and 3. So all you do is you go back, session 22, question 2 and 3, and you review it. Now let's look at the question. Why would beta oxidation occur in the mitochondria? And it's, I think that's one of the silly questions I've seen, where all the enzymes are. Does that make sense? Because it asks the questions and it answers the question as well. It occurs where all the enzymes. I think I do as well. So it's where the enzymes for beta oxidation are located. Is that okay?
Yes.
And how do we get fatty acids into the mitochondria? Anyone know? Okay, we probably could see it. First,
Malleate?
Pardon?
Is it malleate? Oh, my brain's thinking of it.
You first have to activate it, and then the carnitine transport. Yep, I think it's right here. Before can be used in a bit of fatty acid transfers first to CoA, and then carnitine transport. So it's right there. Does that make sense? So, okay, let's go through step by step so everyone is clear about this. We know why it occurs in the mitochondria, because the enzymes for beta oxidation are located in that spot. What is the input for beta oxidation and what is the final output? Who knows that one? What's the input?
Fatty acids.
Fatty acids. And what's the final output?
Acetyl-CoA. Acetyl-CoA.
Yeah, what else do we get out of it apart from acetyl-CoA? These two things here.
Oh, the NADH and FADH too.
And FADH. Excellent. So if you do that, it means that you can get the other 2 marks there. Acetyl-CoA. Where does the input required for beta oxidation come from? Where do we get the acetyl-CoA for beta oxidation?
Lipids digested.
Yes. From lipids. Yeah. Fatty acids. First, the polycysts to generate fatty acids, and then we split the fatty acids into 2, 2, 2, 2. Yep. Okay. Any questions so far? Who thinks the exams will be difficult? Yes?
No, I think
Or
it's,
who?
Hopefully not.
I think, yeah, I don't think it's that they are difficult. If you have done the assignment, it's very similar to the questions you've answered. And these are exactly what the questions are. There are a few things that it's not clear. For instance, if you are asked, what happens if you cannot metabolize carbohydrate? What would happen if you cannot metabolize carbohydrate? What do you think will happen? This is a general information. If someone cannot metabolize, what do we get from carbohydrate, that metabolism?
Glucose.
Glucose, and if you can't metabolize carbohydrates, then you'll have a high level of glucose within the blood.
Yes. When we say carbohydrate, if you cannot metabolize it, it means that you will not, you can get the digestion part, but it will not go into the cells for what? The chemical reaction to take place.
Oh, so that's diabetes.
Then, yes, you can. If it asks you, if you cannot, what would be the outcome if someone cannot then the person is insulin resistant. That's the reason. But in
Insulin
case you have insulin resistance and therefore you cannot get glucose into your cells to break it up, it means that the energy from glucose, you're not going to get it. Does that make sense?
Yep.
Number two, you are not also not going to get pyruvate. And if pyruvate is also responsible for oxaloacetate, if you remember, if you have seen that in your notes. Where do you remember oxaloacetate?
Citric acid cycle.
Yeah.
Yep.
You need it for the citric acid cycle. Therefore, if you don't have oxaloacetate, you cannot get what? You cannot do the citric acid cycle efficiently. Because it's, yeah. And then what happens to acetyl-CoA if you cannot do citric acid cycle? Do you still remember that one? If it's channeled into what?
Oh, fat.
Ketogenesis. If you have a lot of acetyl-CoA and you are not getting it through the ketone bodies. And so that one you have to just think through that. Is that okay there? But it's also in the tutorial answers as well. Question number 3, question number 4. Now, this question, what modification is required so that fatty acid can be used to start beta oxidation? And everyone knows there are 2 things. One, we have to activate it by attaching what? Coenzyme A. Is that okay for everyone?
Mm-hmm.
Yeah.
Yep.
And number 2, once you attach it, we need to transport it from the cytosol. Remember, fatty acids have been lipolyzed in adipose tissue. They have been put into the blood, and so if they enter the cell, they will enter the cell into the cytosol first. And then from the cytosol, we need to put it into the mitochondria where the enzymes for beta oxidation are. And therefore, we need a transporter. And this is the
Carnitine. The carnitine shuttle, yeah.
Transport. I know some people buy it. I'm not sure what it does for them or whether they are not producing adequate amount.
Gym junkie.
Any questions? Okay. Maybe that's what, if you need a lot of a quick burst of energy, then it will help. The next question is, look at this question carefully and tell me are there 2 questions in one or just 1 question? The 5.
2.
2, yes. So sometimes a student will read the first one. What is the final output of beta oxidation? And then everyone knows final output of beta oxidation is what? Acetyl-CoA. And leave it as that. And then someone will go on, and for what purpose? What's acetyl-CoA used for?
To make ATP.
Yes, but first, well, you have to know exactly where it goes.
So aerobic respiration or ketogenesis.
Acetyl-CoA goes through the citric acid cycle to generate NADH, which will be used for what? ATP. So make it as simple as possible and in order. If you have acetyl-CoA, you know that's where it goes. And the reason why it's going there is to get us NADH and FADH2. And what for? For ATP. Does that make sense for everyone? And if you are not sure, just go through this notes. Yes, acetyl-CoA, citric acid cycle. So you mentioned that. And from there, what do you get? You get NADH that goes to the electron transport chain to generate finally what? ATP. Everyone's clear on that one?
Yes.
Yep.
Okay. Next question. Why is it necessary for us to perform beta oxidation? Why is it important? We perform it so we get what out of it? Acetyl-CoA from fatty acids, and so we can get ATP from it. So just be basic. Why are we doing beta oxidation? Acetyl-CoA, we can pass it through the citric acid cycle and ultimately get ATP. So if you like, anytime they ask you, just say that. Why do we do glycolysis? So we can get pyruvate out of it, make it acetyl-CoA out of it, take it through citric CAC, etc. ATP.
Mhm.
Yep.
Excellent.
Why do you, something like that? In that, so it's clear that you know exactly what happens. It goes through here, and the reason is that. The final reason is ATP. It goes through the cycle, it generates NADH, and the final reason is ATP. Any questions? Everyone's clear on that one?
Yes, thank you.
So we get ketogenesis, or which one? I think we're not going to have a lot of time for all of them. So I want you to select the one that you think you probably need help with. So we can go through. We only have 10 minutes left.
Can I just ask, are we going to get, sorry, are we going to get the results for the assignment before the exam?
Usually it's yes, I think you would.
That would be
But
helpful.
most people have done very well, so don't worry too much about it. Yep.
Okay.
You'll get it before. Any of these questions, so we can go to it and have a look at it, that you think you need help with in 10 minutes. Caroline?
Can we go over the keto acids and carbon skeletons?
That's what I was just about to say.
Please.
This one?
Yes. Yes.
The using keto in okay. So session 24.6. Let's go through, let's look at that question. Session 24. The first thing you have to remember that amino acids, there are two parts to an amino acid. The amino group and the carbon skeleton. What was the question? Was it? What was the
number 6? Question 6. It was 24.6.
24.6. Okay. 24 question 6. Need to go down further. Question.
Also, another quick question, Eric. Are the questions selected at random for the start? For question 7, are the questions selected at random or for the topics?
No, it's exactly the same questions you have been giving. So if you learn them, it would usually start with enzymes first, and then second you come to glycolysis and gluconeogenesis or and citric acid cycle, and then you end up with protein. The questions on protein, just like you are asking now.
All right.
Yeah.
Excellent.
So just follow that, and you'll be able to do all of them. So after an amino acid has lost its amino group through transamination reaction, all that remains is the carbon skeleton, which is a keto acid. What is the difference between glucogenic and ketogenic amino acid? And anytime you remove, or I would say you yank off the amino group of the carbon skeleton, some of the carbon skeleton, it can be pyruvate, or it can be acetyl-CoA, or it can be acetyl-CoA. If the carbon skeleton is a pyruvate, then it means that you can get glucose out of that pyruvate in gluconeogenesis. Does that make sense? Then it means that original amino acid was a glucogenic amino acid. On the other hand, if it gives you acetyl-CoA or acetyl-CoA, it means you can get ketone bodies, because to synthesize ketones, you need acetyl-CoA in acetyl-CoA. So you call that kind of amino acid ketogenic amino acid. Does that make sense for everyone?
Yep.
Okay, question number 2. What kind of chemical reactions would glucogenic amino acid? And straight away, I think a lot of people get this wrong as well. If you get pyruvate or oxaloacetate, where can you take pyruvate and oxaloacetate? Which reactions?
Citric
can
acid
put
cycle.
citric acid cycle. Yes, you can convert it in. Or what else? Apart from that, what other reaction can you put it in? You did know, you still remember gluconeogenesis, isn't it? It's the non-carbohydrate part of gluconeogenesis. So gluconeogenesis is one of them. That's this one. Because you go backwards to synthesize glucose. And then there's a question as well that I want to ask. Can you get fat by eating too much proteins?
Yeah.
Yes or no? Yes. Why?
Because when the carbon skeleton is used then to produce pyruvate, then that can be put back into the citric acid cycle, and then you get ATP, and the excess can be stored in the adipose tissue.
Yes, then you get the excess acetyl-CoA from it going into what? Lipogenesis, creating fatty acids. Yes, lipogenesis, creating fatty acids, and then being stored in what? As starch in adipose tissue. So remember that one, that you can still get obese by eating a lot of proteins. Everyone's clear on that one? What about ketogenic amino acids? What metabolic reactions can ketogenic amino acids be used for? And then you can say, if it's acetyl-CoA, it means citric acid cycle and what? Ketogenesis. Does that make sense? That if you get ketogenic amino acid, it means the carbon skeleton is either that one or that one. If the carbon skeleton is this one, if the carbon skeleton is this one, straight through what? Citric acid cycle and also ketogenesis. And acetyl-CoA is on the second path, the second point of ketogenesis. So it goes through what? Ketogenesis. You don't have to write the whole lot here. If you understand this, you can say that if it's acetyl-CoA, it will go through the citric acid cycle to generate ATP. If it's acetyl-CoA, it can also be used in what? Ketogenesis to produce ketone bodies. Any questions? Now, one more question before our time is up. So look again and all right. Fed, let's do this. Fed and fasting state. I think that's an easy question to get as well. I think we've done it already. The fed state, what hormone will be released in the fed state?
Insulin.
Insulin, and you just go on and say insulin is released by what? Pancreas. And then the insulin will do to the, yep. And then what about the fasting state? What hormone is released? Will be released from the pancreas. And what would the glucagon do?
Takes the glucose out of the cell.
Yes, it takes out of the cell, from liver cells into the blood. And where would that go? Into
the liver.
Once you produce glucose from gluconeogenesis or glycogenolysis, it goes into the blood, and the blood takes it to cells or tissues that require glucose, especially the brain and then the testes. They just doesn't want to depend on fat. They want to depend only on glucose. I think we've got 2 minutes. So, using fatty acids to generate ATP. If I ask you the structure of ATP, who still remember what is the 3 components in ATP for a chocolate, which I can email?
A nitrogen base, ribose sugar,
nitrogen base,
and 3 phosphates.
Excellent. You write all of those, then you get on marks there. It's consisting of adenine, which is a nitrogen base, a ribose, and 3 phosphates. Is everyone clear on that one?
Mm-hmm.
Now, how do you get ATP from, if you have one NADH, how many ATPs are you going to get out of it when it goes through the compound?
1.5.
NADH.
2.5.
2.5.
2.5. Yes. So make sure you don't swap it around, because if you know it and you swap it around, you still get it wrong. What about 1 FADH2?
1.5.
1.5. So make sure you are very clear that anytime you have 1 FADH2, you get that. And then you are asked, if you have 1 citric acid, you remember the citric acid cycle. How many NADH do you get out of 1 citric acid cycle? 3 NADH and 1 what?
1 FADH2.
FADH2. If you have 3 NADHs and each one of them is 2.5, the total will be how much? 2.5 times?
7.5.
Excellent. And then you have 1 FADH2, you get 1.5. Do you still get another energy from citric acid cycle apart from the reduced coenzymes? You still remember GTP? There is 1 GTP.
1 GTP.
Which is equivalent to what? 1 ATP. So you add to it. And so what is the total ATP you get from 1 citric acid cycle? 7.5 plus 1.5 plus 1 is what?
10.
If you do 2 citric acid cycles, how many ATPs are you going to get? 10 plus what? 10 times 2. Is someone follow this?
20.
20, yes, exactly. All right, I think we've done it 1 hour. And so what I'll do is I'll stop the recording, but you'll be free to still ask questions before we close.